Okay, ma. Okay, so when you observe this vaccination, the vaccination process is mainly based on the memory of B cells and T cells. Okay, that means the vaccines also generate a memory of the T cells that recognize the pathogen quickly and subsequent exposure. That means for the second time. That means okay, when we are giving vaccine means we are giving the inactivated pathogen or antigenic protein, okay, for the first time. So our body will show response very slowly. Okay, that is the first response. But when the actual disease causing organism is entered into our body, now this exposure is considered as the second exposure, and the response shown by our immune system is the secondary response. So the response will be very quick and produce large number of antibodies. This is all because of the presence of memory cells of the immune system, that is the B cells and the T cells. Okay, so as a result, okay, okay, the pathogen when entered into our body, okay, that is a subsequent exposure and overhelp the invaders with a massive production of antibodies. So even a small number of pathogen is entered into our body will show huge response and protects our body from the infection because of the presence of this because of the huge number of antibodies produced by these memory cells. Okay, that is what we'll do in case of vaccination. But in immunization, what we'll do? If a person is infected with some deadly microbes to which a quick immune response is required, tetanus, we need to directly inject the preformed antibodies or antitoxin. So that means if a person is already infected, in that case, the vaccination cannot work because already infection is there. So in that case, we can't give vaccination. In that case, what we can do means, okay, we can give to that person who is already infected. Okay, we can give the ready-made antibodies. That means the antibodies which were already prepared. Okay, those antibodies are directly injected into the body. Okay, and that will protect the body from a further deterioration of the condition. Okay, that is okay. For example, the tetanus injection we are giving, TT, tetanus toxoid we are giving. This is a TT. TT means a tetanus toxoid. Okay, that is the antibodies, the already synthesized antibodies. Okay, in the form of a TT injection we are providing. That is what we called antitoxin. Okay, for tetanus or snake bite. Okay, for all this. Okay, that is such a phenomenon. We are calling it as immunization. So if you are giving ready-made antibodies, okay, to protect, okay, not to protect, to cure the disease, because already the person is infected. Okay, then such a process we are calling it immunization. Okay, that is so we can see even in case of snake bites, the injection which is given to the patients contain preformed antibodies against the snake venom. And this type of immunization is called passive immunization. So this is okay. That means in immunization, what we'll do, the ready-made or preformed antibodies are given to the person who is already having infection or already who is affected. And this process we are calling it as a immunization or passive immunization also we can say, because we are giving antibodies ready made. But when you observe the vaccination, vaccination is considered as a active immunity, because our body itself will prepare the antibodies or produce the antibodies, because as we are exposing to the proteins which represents the antigenic structure or inactivated pathogen, we are injecting into our body. So again, is that our body will prepare the antibodies. That exposure is considered as the first response of the immune system. When the actual disease causing organism entered into our body, because the presence of memory T cells and B cells, okay, our body will show anamnestic response. That means okay, quickly, and a huge number of antibodies are produced, and that one will protect our body from a infection. Protecting here, the protecting, okay, that means before occurrence of the disease condition only the organism is subsided, the organism will be disabled. But in case of immunization, already the person is suffering from certain infection. In that case, we are giving the ready-made antibodies. Okay, and that phenomenon we are calling it as a immunization, or in other terms, we can also call it as a passive immunization. And next, okay, how this okay, we can, okay, in case of vaccination, we are giving inactivated pathogen or weakened pathogen. That one we can do by heat killing or by using certain chemicals we can make. But how to produce the proteins which will represent the antigenic structure of the pathogen? Okay, we can't produce from the microorganism, okay, that disease causing organism, okay, because they are present associated with that pathogen itself only. We can't able to okay obtain separately, because they are completely associated with that disease cell. Then how we can able to obtain these antigenic structures, the proteins which represents the antigenic structure? Okay, and that is achieved with the help of recombinant DNA technology. Okay, the recombinant DNA technology has allowed the production of antigenic polypeptides of pathogen in bacteria or yeast, whether it is a whether the pathogen is a bacteria or virus or fungi or anything. Okay, okay, whenever this disease causing organism is exposed to our body is producing antibodies, but these antibodies are not produced against the total organism. It is produced against the antigenic structure that is associated with that pathogen. Okay, that antigenic structure might be a protein or a carbohydrate or a lipid or any other. It might be anything it might be. It might be a protein, or it may be protein in nature or carbohydrate in nature or lipid in nature, whatever it may be, whatever the nature it might be. Okay, what they will do, they will identify that antigenic structure. Okay, for producing that antigenic structure, one gene will be there which is associated with the genetic material of that pathogen. Okay, they isolate that gene, and that gene which is present in that pathogen which is producing that antigenic structure, that is introduced into another organism's genetic material like bacteria or yeast. and now this bacteria or yeast are allowed to grow. When it is growing, it will produce that antigenic structure. Understand? So that means in every disease causing organism, some some antigenic structure is there. Okay, to which against our body will produce antibodies. Okay, so in the pathogen also, for the production of that antigenic structure, some gene will be there in the genetic material of that pathogen. The sign, this biotechnologist, what they will do means they will identify the gene which is involved in the production of that antigenic structure. Okay, it may be the protein or a lipid or a carbohydrate, whatever it may be. Okay, they identify that gene, and now that the gene is isolated from that pathogen and is introduced into the genetic material of any organism which can be easily grown, like bacteria or yeast. Okay, introduced into the genetic material of this bacteria or yeast, and this yeast or bacteria is allowed to grow, multiply in number. Okay, and it is provided with a certain medium so that the gene which is introduced is allowed to express, and that antigenic structure is obtained. Whatever the antigenic structure that is produced, that is isolated and that is given to our body in the form of vaccine. Okay, that is a recombinant vaccine. Such vaccines we call it as recombinant vaccines or second generation vaccines. Remember, okay, these vaccines which are produced by using biotechnological process or recombinant DNA technology, we are calling it as recombinant vaccines or second generation vaccines. Then what is the first generation vaccines? The first generation vaccines are the vaccines which are produced by inactivating attenuated pathogens. Inactivated or attenuated. Okay, if you are preparing the vaccines by using inactivated or attenuated pathogens, then such a type of vaccines we are calling it as a first generation vaccines. These are the generations that were developed by the Edward Jenner and later on by large number of scientists. But nowadays we are not developing this first generation vaccines because they are sometimes harmful. That's why, okay, we are using recombinant vaccines. Okay, these are more safer because here no pathogen, only the antigenic structure we are giving. Okay, as we are giving inactivated or okay, sometimes it may become active and they may cause disease. That's why some side effects are there. But in recombinant vaccine, as we are giving only antigenic structure, we are not giving the total organism, whether it is inactivated or attenuated. We are giving only antigenic structure. That's why the recombinant vaccines are much safer when compared to other, the first generation vaccines. Okay, and next, the vaccines produced using this approach allow large scale production and hence greater availability for immunization. So, and next, when you observe, okay, the vaccines which were developed by recombinant DNA technology, okay, by using this process, we can able to produce, large number of vaccines within a short period of time. That's why, actually, we are getting the corona vaccine, different types of corona vaccines. Actually, for preparing a vaccine, it will take some 5 to 10 years. They have to undergo different trials. according to the old method, that is the first generation vaccines. But with the help of the new this method, recombinant DNA technology, okay, we have got this vaccine within 1 or 1.5 year. Within 1 year, we have got the vaccine. And the immunization program is going on. This is all because of the recombinant DNA technology. So, okay, we can able to get a large scale production of vaccine. And next, and it is availability is also more. Okay, the first vaccine that is produced by using this recombinant DNA technology is a hepatitis vaccine produced from yeast. Okay, hepatitis B is a virus. Hepatitis B is caused by a hepatitis B virus. But but the vaccine for the hepatitis B virus is produced by using yeast. Here what they are doing, the antigenic structure which is representing this hepatitis B virus is produced by introducing into the yeast cells. Okay, that is very okay, and we'll also discuss some more related to this recombinant DNA technology while we are discussing about biotechnology in the 11th and 12th chapter. Okay, then you'll understand better. Okay, that is about this vaccination and immunization. Anybody is having any doubt?
No, sir.
Okay, ma'am. And the next one is allergies. It is also a immune response. Allergies are also a type of immune responses. That's why we'll discuss this allergy under immunity only. Okay. Generally, when you have gone to a new place and suddenly you started sneezing, wheezing for no explained reason, and when you went away, your symptoms disappeared. Okay, that is when you are okay, or even sometimes even when you take some types of food, okay, which is not good for your health, immediately you start sneezing, wheezing. That means okay, some kind of disturbance in your body, the food material, or when you go to new place, or when you go to hilly regions, or or some other places. Okay, suddenly without any reason, here we don't have any proper reason. Without any reason, it will be you start sneezing continuously, the nose will be draining and wheezing. That means you start okay, some disturbance in your body without any reason. Okay, did this happen to you? Okay, that means you might have, okay, in your everyday life you might have observed this thing, okay, now and then. And some of us are sensitive to some particles in the environment. Some of us are sensitive to some particles in the environment. That means some persons, we can see some persons cannot go to the ground and they cannot play in that dust. Okay, they used to play indoor, mostly games, not sports. Okay, they used to play indoor with well placed, but they will not show interest in going out. Why? Because because of they are sensitive to some particles in the environment. So above mentioned reaction could be because of allergy. Okay, and already we have discussed while we are discussing about sexual reproduction in flowering plants, while we are discussing about pollen grains. Okay, some pollen grains are allergic, like parthenium. Okay, the pollen grains are allergic to. Okay, allergy to pollen. Okay, allergy to mites. etc., which are different in different places. Okay, some are showing the allergy to pollen grains, some are showing allergy to some insects, some are showing some allergy to mites. Okay, like this, they are showing a different ways of allergy. So what is mean by allergy? Okay, what is mean by allergy? The exaggerated response. The exaggerated response of the immune system to certain antigens present in the environment is called allergy. That means so you have to remember that allergy is also caused because of the antigen. So the exaggerated means our immune system will respond quickly, huge in a small period of time. Okay, quick and a huge response in a short period of time. That's what we call it as exaggerated response. The exaggerated response of the immune system to certain antigens present in the environment is called allergy. So whenever any dust particle is entered into our nose, okay, we'll suddenly start sneezing, wheezing, and all that. That means, okay, in the nose, it is showing some exaggerated response, our immune system. So this exaggerated response of the immune system to certain antigens present in the environment, we are calling it as allergy. So the substance to which such an immune response is produced are called allergens. So that means because of certain antigen, this exaggerated response is shown now. Okay, whatever the substance to which this exaggerated response by the immune system is shown, okay, that substance we are calling it as allergen. Okay, and next, okay, what are the antibodies that are mainly responsible for this allergic reactions or allergic responses? Means that is nothing but IgE antibodies. Already we said no, there are different types of antibodies: IgE, IgG, IgM, IgE. In that, okay, IgE antibodies are mainly involved in allergic reactions. So the antibodies produced to these are IgE type. So the IgE type of antibodies are responsible for this immune responses. Okay, common examples of allergens are mites in dust. Okay, what are the common examples of allergens means? Okay, mites in dust, pollen in the air, and next animal dander, that is the dust that is produced from the dried cow dung, that is called animal dander. When the animals are moving, okay, if a group of animals are moving, herd of animals are moving, okay, okay, because okay, that means as they are moving, okay, in together, a large amount of dust from their through their legs are produced, and these dust also a type of allergen. Okay, that is so mites in dust, pollen, animal dander, etc. Because everyone will be having a different allergens. Even some members are allergen to certain oils, chemicals. Okay, some food materials. This is all. Okay, how they can identify the allergen? Okay, how they can the allergic reactions. Okay, symptoms of allergic reactions include sneezing. Okay, that is a running nose, sneezing, watery eyes. Okay, okay, the eyes are produced from the eyes. Okay, sir, water is produced continuously from the eyes with no reason. And next, running nose and a difficulty in breathing. Okay, the person finds a difficulty in breathing. So these are all the some of the symptoms for allergic reactions. That is sneezing, watery eyes, running nose, and a difficulty in breathing. Okay, and next, allergy is due to the release of chemicals like a histamine and from the mast cells. Okay, that is, and next, how these responses are initiated means whenever the allergic substances or allergens are entered into our body, okay, our tissues will produce certain chemicals like a histamine and it is serotonin. Okay, from a special type of cells that are present in the tissue called mast cells. Okay, a special type of cells called mast cells are present in the tissues, and these mast cells will produce two chemicals. That is, one is histamine and the second one is serotonin, and these are responsible for this allergic reactions primarily. So for determining the cause of allergy, then how we can able to identify a person is having allergy to a certain substance. For determining the cause of allergy, the patient is exposed or exposed to or injected with a small doses of possible allergens and their reactions studied. Even you can see sometimes when you go to the hospital, okay, whenever you want to give certain antibiotics, okay, or certain injections which are a little bit allergic, then first of all they'll give on the hand. Okay, they'll give to the skin of our hand, and they will okay, leave it for about 1 hour or 2 hours for observation. Okay, and they will check any allergic reactions are shown by our body. In our hand also tissues are present, and in that also the mast cells are present. So if that chemical is a allergen to our body, they show immediate response in the form of allergy, some rash like or some bubble like, okay, something change will be observed. Or if there is no okay, if it is not causing any allergic, no reaction, it will be normal, like normal injection. Okay, then they will give you the dose of that injection to your body. So that is so the possible whatever the chemicals that are possibly causing allergy, those chemical substance are first they are tested on our body. Okay, and they will check for the reactions. Okay, sometimes we can say no. Okay, I sometimes they say okay, I'm allergic to certain antibiotics. I am allergic to certain chemicals. I am allergic to certain foods. How we are able to know? Because they are all tested. Okay, they are given that type of food, and they will observe the reactions. Any changes are taking place in the body. Okay, by which they can identify the allergen. So, for determining the cause of allergy, the patient is exposed to or injected with a very small doses of possible allergens, and the reactions are studied. So that means it will be exposed. For example, if you want to know if the person is allergen to certain pollen, the person is exposed to that pollen grains. For example, if you want to know the whether the okay, for example, okay, for diabetic patients, they will give insulin. Okay, there are different companies of different types of insulins. So before they are giving the insulin for the first time, first of all, they will test it, whether that type of insulin is working normally or not. That means that they will check for any allergic reactions, the insulin, before giving to the person. So they'll give it to the skin first and they will check it. So small doses are given, are injected into the body, and they will check for the reactions. So that means we already said what are the chemical substances that are mainly responsible for allergic reactions means histamine and serotonin, which are produced by the mast cells. So, okay, if any allergy is observed or if any allergic reactions are observed in any person, how we can able to reduce this allergic reaction? We already said it is because of the release of the certain chemicals like histamine and serotonin. So we can give antihistamine drug. So the use of drugs like antihistamine, adrenaline, and steroids quickly reduce the symptoms of allergy. So that means we have to give certain chemicals which will reduces the production of this histamine and serotonin. Okay, like antihistamine or adrenaline, and also this steroids. So there are some steroids, okay, which will reduce the symptoms of allergy, so that we can protect that person. Because okay, if the allergic reactions are prolonged for a longer period of time, it may lead to the death of that person also. Okay, it may lead to the death of that person. So okay, we have to make sure that we have to reduce that allergic reactions. For that, we can use this okay, some of the drugs like antihistamine, adrenaline, and steroids, which will reduce the symptoms of allergy. Okay, somehow modern day lifestyle has resulted in lowering of immunity. Okay, somehow modern day lifestyle has resulted in lowering of immunity and more sensitivity to allergens. And more and more children in metro cities of India suffer from allergies and asthma due to sensitivity to the environment. Okay, when you observe here, those children, especially who are living in cities, usually they will not come out of that, okay, in the young age, okay, because they do not find that much place for playing outside. Okay, they will be completely in indoor, so they are not exposed to different environmental conditions. Okay, when they suddenly comes out of the houses and they move to different place, as they are exposed to different environmental condition, okay, because of the sensitivity that is developed by that person because of continuous indoor living, okay, he will be okay whenever he goes to outer environment, okay, that environment, whatever the substances that are present in that environment will become as allergens, and they cause allergic reactions. In the same manner, in case of metro cities, okay, because of pollution, a large number of chemicals are released. Okay, because of this also, okay, the sensitivity is reduced, okay, and the immunity is also at lower levels because of continuous exposing to certain chemicals, and they will become more sensitive, and they'll show allergic reactions very quickly. That's why, okay, we have to allow the children to expose to different environmental conditions in the young age itself only. then only they can they get exposed to different environmental conditions, so that they can develop immunity, and they develop less sensitivity to different substances, because they are continuously exposing with a different environmental conditions, no, so they develop certain immunity, and they themselves they can protect against certain disease causing organisms. This could be because of the protected environment provided early in life. So that means why this why the children that are present in metro cities are developing more sensitivity, and developing allergic reactions for when they are exposed to different environmental conditions, means because in the early days of their growth, that is in the young stages or in the infantile stages, they are not exposed to different environmental conditions. As they are growing in a protected environment, as they are protected environment, they become sensitive to different substances and show allergic reactions when they are exposed to different environmental conditions. That is about this allergy. Anybody is having